when it comes to bioethics, like who has to agree to what, and like what, is it by country? Is it by and like what potential like dangers are there, or like gray areas
Well,
of it?
something that is really controversial right now, specifically these days with CRISPR, is that it's just like, let's say we genetically modified, like let's say a human newborn, just to be smarter. I don't know how is that possible, but people will be like, oh well, he has an advantage on us because like, he was genetically modified and he's gonna be smarter than us, and that can cause some discrimination upon us.
So something I'm curious about is, like, right now when we talk about discrimination, it's a lot about skin color, ethnicity. Will there be a point where the discrimination is against like, it's like genetically modified humans and then like non-genetically modified, like
Sadly,
born
I think
humans?
so. Yes,
Yeah.
sadly, I think so. Yes. Yep. Specifically for like for job appliances, let's say, someone who has like this syndrome or these problems, and this guy was born with the same syndromes and same problems, but he was CRISPR, so he doesn't have them anymore. He's gonna get a job. And that takes into disadvantage that people who actually have like these diseases they're not able to afford it.
What's the cost? You said it's cheaper to do it now. Like, is it so cheap? Well, I don't know.
Well, to give you an idea, like I think in my lab I spent around $100 just to get gDNA, which I actually make it sgRNA.
I have no idea what that means.
So it's basically you have like a template of DNA, and from there you make RNA, and you keep that RNA and you use it as sgRNA, so the Cas9 can go to the gene of interest you want.
So you did this stuff for $100,
basically. But after that, you need to have like the whole machinery. You need to do a lot of sequencing. You need to take a look into the mutations. Then you need to take a look if like the Cas9 will cut in the sgRNA where your genome is.
So
because
yeah, go ahead.
Oh, sorry. So one of the questions I actually have up to this moment with CRISPR is like, okay, the sgRNA is only 20 nucleotides long, right? 20 base pairs long. So you're trying to target a sequence of interest. So how do you make sure that this sgRNA is going to target your sequence of interest and not other sequences? Because it's like you can have the same sequence in another gene that encodes for another thing, and you actually do not want to knock out that gene. So that's my main question here with CRISPR. Like, it's like, are you sure that this sgRNA is gonna bind to your gene of interest and not to another area you don't want to?
Has that been tested yet?
I'm not quite sure, to be honest. But in part of my research, actually, I'm trying to send everything for sequencing. I'm gonna have the results pretty back soon, so I'm gonna have like some results.
And what about in
I will
humans?
be able to see. I got no idea how can that be applied in humans, to be honest. Like, there's like another topic in CRISPR which comes to primer design. That's another topic that's not like my specific area, but as more accurate your primer is, the more accurate your sgRNA is gonna also bind to the sequence of interest.
So we haven't tested this in humans yet, so we don't know for sure, but theoretically, this is
it's possible. Yes.
Yeah.
This is like the stuff of the future. That's 100% sure. Some people, I'm like confident that they're trying this technology in humans. Like some scientists, I'm 100% sure that they've designed an sgRNA like with a specific binding they want, so it doesn't go anywhere else.
So this is really like a turning point in the human story in a way, right?
Yep. like back in the 1940s, 1920s, like people were, it was physics, because people were like, oh, the existence of molecules, or they were just discovering the elements. And now we have CRISPR in biology, which is actually a tool that can solve like health problems. So this is basically like, this technology is just born, so it's just going to evolve over time.
And now with like the way the 21st century has, well, I mean, technology in general, but everything is compounding. All the technologies are getting better and better, quicker and quicker. And it's not going to stop, of course, on that pace. And this is like a profound thing that can impact the human phenomenon.